Right, pnawn da, hello everybody, my name is Mr Bryn Davies and I will be doing the A-level biology revision session today on sexual reproduction in plants. That is unit 4.2, not as the title says 4.1. Okay, so that's 4.2. Please forgive any linguistic slip-ups. My first language is Welsh, so some of this may be a little bit stilted, but here we go. Okay, here's a list of the sessions that Cadwell Cymru have recorded this spring. The AS and A-level biology sessions are all there. One's on cell structure and microscopy, 1.2. And classification and exam technique there for AS, and then ATP synthesis and chemiosmosis and mitochondrial membrane proton gradient, so the respiration bit for unit 3, and the human and plant reproduction for unit 4. Okay, the aims of this session is to recap the subject knowledge for the plant. Pardon me, reproduction units. Okay, if you have a preferred note-taking method, then please continue that. If not, I'd like you to try this Cornell method of taking notes. Okay, so if you divide your page down by a third like that, okay, and then have a section at the bottom. So the main notes that you're going to take from what you see in here today are going to be in this bit. These other bits here you do after the presentation. Okay, so this bit here, questions you might have about what you've written, okay, some prompts to remind you about it, and then the summary here, so you're condensing all of this down into a brief summary. That is a very good beginning to your flashcards if you make flashcards. Okay, when you are doing your notes here, okay, be efficient, paraphrase, yeah, think of short, punchy things to say as opposed to writing out lots of very laborious notes that you might have, okay? Excuse me. So here is the syllabus points for 4.2, sexual reproduction plants. It does appear to be quite a short one, yeah, with only seven sentences, but there is, as I'm sure you all know, some complex bits in the A-level biology. Okay, there's also some specified practical work, okay, and we'll try and go through that as well when we get to it. As well as these Cadwell Cymru videos, there are also some very good resources available on the WJC website. Okay, so if you go to the WJC website, A-level biology, okay, there are past papers and mark schemes there for you to go through. Okay, the key documents, the syllabus is there, the teacher's guide is there. It's all very good. Okay, if you also Google WJC blended learning, okay, you can find the blended learning resources for this unit. Okay, the blended learning that WJC have put together is really good. Okay, so it's it's a very good extra on top of your lessons that you're having with your teachers already. There are other resource sites out there, but less is more. Okay, you need a few high quality revision resources is an awful lot better than loads of rubbish ones. Okay, so don't keep going to find new ones, going to find new ones, going to find new ones. Okay, stick with some good ones. Okay, good job. Right, so we will start as with most things in biology, there is a structure and function bit. Okay. So there are 4 generalized bits to flowers, which are the reproductive organs of plants, in case that is something we need to say. Okay, so flowers. Okay, so in angiosperms, okay, so angiosperms are flowering plants. So flowers are the reproductive organs. Okay, so here is a generalized structure of a flower, and there are 4 different types of adapted tissues or leaves, really. Okay, you have the carpel, okay, which is the female part. You have the stamen, which is the male part, and then you have two rows of petals or adaptive leaves on the outside. Okay, so the calyx is the outer ring of sepals, these. Okay, the corolla is basically the scientific word for the petals, so these. Oh, pardon me. Okay, the stamen consists of three parts: the anther, the bit at the top that produces pollen, the filament that holds it. Okay, so they are the two bits there of the stamen. Okay, so the stamen is those two bits there. Okay, and these are the male parts. These are the pollen producing part. Okay. The carpel, okay, is the female part. Okay, so the carpel consists of the stigma, okay, the style, and the ovary at the bottom with the ovule. Okay, so the stigma is a sticky bit at the top where pollen sticks to. The style then is a tube that leads down to the ovary. Okay, so the ovary there has lots of ovules in it. Now, each of these ovules, that is what will develop into a seed. Okay, so if you could draw some comparisons with the human reproduction. Okay, so the female ovary contains ovules. Yeah, so a similar sort of thing. It's the ovule that will develop into a baby. Okay, the ovary is the receptacle, the receptacle that holds those ovules. Okay, you also have a receptacle. So this is the bit that all of those petals, all of those adapted petals joins onto here. The sepal is, says there's not a line for the sepal. So the sepal is the bit that closes around the flower bud to start with. Okay, and some flowers, depending on whether they are insect or wind pollinated, can at the bottom here have something called a nectary. Okay, now the nectary is round at the bottom here, and as the name suggests, it will produce nectar. All right, and we'll talk about why it produces nectar when we talk about how pollination occurs. Okay, so that's the generalized structure there. There's lots of learning stuff there. Okay, so that's something that you need to review again and again until you've got the structure and the function all sorted and square in your head. Okay. Right. Wind pollinated flowers actually look very different. Okay, so wind pollinated flowers don't have the brightly coloured petals. They don't have the nectary. They don't have the nice aroma that the insect pollinated flowers do. Okay, so they look very, very different. So we'll talk just here a little bit about these. Okay, so wind pollinated flowers have small petals or no petals at all. Flowers are located above the leaves. Okay, so there's good access to wind. Yeah, produced early in the spring to give maximum advantage to wind currents without interference from the leaves. Okay, so they often produce lots of pollen early on in the year. The stigmas are large and feathery and hang outside the flower to catch the airborne pollen. Okay, so instead of having an insect pollinated flower that's got petals like this and then a tiny little stigma inside, what you will tend to have is a little flower, but you'll have a big feathery stigma like this. Okay, so any pollen that's flying around in the air will be caught on that feathery stigma. Okay, again, large anthers suspended outside the flower that release pollen grains. Okay, so again, you've got these pollen grains that are released from large large anthers on these stamens. So they're again released out into the wind currents. All right. And as you might imagine, vast numbers of pollen, loads and loads and loads of pollen. So trees, this is trees are wind pollinated. Okay, so they produce copious amounts of pollen, which is what gives you hay fever. Okay, these are different to insect pollinated plants. Okay, so insect pollinated flowers have a different mechanism. Okay, so instead of just throwing pollen out into the air to be carried to the next flower by the wind, they depend on insects to carry it for them. Okay, and there's a symbiotic relationship here between the insects and the flowers, all right, between the plants. So large, brightly coloured petals, often with guidelines to the nectaries to attract insect pollinators. Okay, it's very interesting to look at flowers under UV light. Okay, so some flowers will glow under UV light. Insects are capable of seeing UV light. Okay, so it's basically just like a big runway. Okay, these insects are drawn into these flowers to get their pollen. Okay. Yeah, that's misspoke there. So the insects go after the nectar, okay, that they will then use, bees will store it, turn it into honey. Okay. Pollen is kind of like a byproduct for the insect, really. The insect will go to the next plant and it will fertilize the next flower, which is an advantage to the plant, obviously. Okay. But it's not actually sometimes the pollen that the plant is after, that the insect is after. The stigma, this bit at the top here, okay, is sticky. So when an insect pushes in down here to get to the nectary, it will push past that stigma, and any pollen it might have on its body will get stuck onto the stigma. Anthers rigid on filaments inside the flower where they must rub against them. Okay, so your anthers are going to be like here. Okay, so when an insect, I can't draw an insect here, but when an insect pushes down here, it's going to rub against that pollen. It's going to rub any pollen it's taken from a previous flower onto that stigma, isn't it? Okay, pollen grains are large, spiky walls to stick to insect bodies. Okay, so there's two mechanisms here of of pollination. Okay, so again, a comparison can be drawn. Pollination similar to fertilization, the transfer of a male gamete onto the female organism. Okay, large sticky pollen, heavy pollen, and less pollen as well on insect pollinated flowers. Okay, so there's again a comparison to be drawn, insect and wind pollinated flowers. Okay. Right, the development of pollen. So this here is a cross section of an anther. Okay, so this vascular bundle in the middle. Okay, the stamen comes up here, so we basically have a bird's eye view of the anther here. Okay, and these grains in here, these are pollen grains. Okay, so each of these is a pollen grain, and that pollen grain is basically the flower gamete. It's the male gamete of the flower. Okay. And you need to be able to identify or have seen a picture of this down the microscope. Okay. Often lilies are used. Okay, so lilies, big flowers with very large anthers. Okay, and the pollen from lily anthers will stain. It will stain, clothes or curtains or things like that. It will stain fabric. But they're very good to look at under the microscope. Okay, so you can see this anther here has 4 pollen sacs. Okay, kind of looks a little bit like a butterfly. Yeah, if I was to draw 2 little antennae here, yeah, it would look a little bit like a butterfly. Okay, so we've got a vascular bundle in the middle. So if you remember from AS, vascular bundle, that's where products of photosynthesis will travel up. Okay, so it's active process here, requires energy, products of photosynthesis in order to form these. Has a fibrous layer on the outside to protect. It has an epidermis, obviously, and it has a pollen sac. Okay, so the pollen sac is this like darker green layer here, and that is where you have the cells that begin to divide to form the pollen. Okay, so yet again, you could kind of draw another comparison here. Okay, so this is the male gamete produced. So in humans, that would be the sperm. So in flowers, it is pollen. Okay, so this pollen sac would be kind of the same as the germinal membrane in the testes, yeah, in the seminiferous tubules. All right, so contains a large number of diploid mother cells here, and these are your pollen grains, which are just N. Yeah, the mother, don't know why I've got a line there. So your mother cells are diploid, okay, and the pollen grains there are haploid. Okay. You then have an area of dehiscence, okay? So the area of dehiscence is where it breaks apart, okay? So it will flip out here and flip out there and release the pollen, okay? So I think I've misspoke there as well, sorry. So this entire thing is called the pollen sac, okay? That layer of cells around the inside is called, there it is called the tapetum. Okay, so the tapetum would be similar to the seminiferous tubules, the wall of the seminiferous tubules in the testes. Okay, so that is the layer there that produces these haploid gametes. Okay, good job. Right, so again, nice, fairly long, complicated process here to produce pollen. So there are similar steps in the production of in spermatogenesis, okay? But in pollen formation, you have a diploid mother cell that is in the wall of the tapetum. That undergoes a meiotic division to produce a tetrad of 4 cells, okay? So one meiotic division, okay? Each haploid cell from tetrad undergoes mitosis to form a pollen grain, okay? So we have a meiotic division and a mitotic division, and then we have a specialization step, okay? So these, this is 2N, these are haploid N, and this is haploid as well, okay? And what we end up with here is the mitosis occurs, and so mitosis as opposed to cytokinesis, okay? So mitosis where the nucleus divides into 2, all right? But the cell doesn't divide. So you end up with 2 types of nucleus, okay? So you end up with a tube nucleus and the generative nucleus. So both these nuclei are haploid, okay? So in insect pollinated plants or flowers, it will produce a thickened layer around the outside with hooks possibly that are going to stick to the insect. All right, wind pollinated flowers, this bit will be much thinner, it will be much lighter, and it will be much smoother. Okay, so again, you can possibly draw comparisons with the process of spermatogenesis, meiotic divisions, mitotic divisions, what happens when. Okay, good job. So the development of the ovule is slightly more complex. Okay, so remember this is inside the ovary. Okay, so your ovary, if I draw now, so there's a stigma on top, that's the style. You have the ovary, and inside the ovary you have these ovules. Okay, so this ovule is basically this. Okay, so the ovule in the middle contains a mass of cells that calls it nucellus. Okay, so this is the entire ovule. So the nucellus is there in the middle. Okay, it has 2 protective integuments around the sides. They are layers of tissue that are going to protect this. All right, that little stalk there joins it to the middle of the ovary. All right. So one cell inside this nucellus has a signal, genetic signal, and that develops into the megaspore mother cell. Okay, so that's a diploid cell there. Okay, so we have a meiotic division. Okay, again, and that produces 4 haploid megaspores. Okay, so it isn't it isn't an ovule just yet. Okay, so it isn't the female gamete just yet. They're known as megaspores. Okay, 3 of these will degenerate, okay, leaving us with 1. There is then a mitotic division, okay, and this will form an embryo sac, okay. So 1 haploid megaspore develops into the embryo sac, so you then get 3 mitotic divisions occurring, okay. So 2 goes to 4, and 4 will then go to 8. All right. So this bit's quite confusing. Okay. So again, this is all happening within the megaspore cell, but there are 3 mitotic divisions of the nucleus. So we end up with 8 nuclei. Seems a bit greedy to me, but there you go. Okay. So there's 8 nuclei. Now, what these nuclei do is they migrate to different regions in the cell. All right. So you have 3 nuclei that will migrate to this end of the cell, 3 that will migrate to this end of the cell, and 2 that will stay in the middle. Okay, so you have 3 antipodal cells there. Okay, so these nuclei that are the result of the mitotic division, will develop into 3 cells here. Okay, the 2 in the middle will develop into something called polar nuclei. Okay, their job will come apparent when we talk about fertilization. And then down at the bottom here, you have the female gamete, okay, and then 2 what are called synergids. Okay, so the pollen that was formed from the previous slide is going to fertilize this guy here. Okay, and there are other jobs for those to happen. But again, all right, you need to be aware and have a knowledge of what's happening here. So a meiotic division and 3 mitotic divisions, and then the structure there of the ovule. Okay, so this is the ovule. This is the female gamete that's going to be fertilized. Okay, it's quite a complex idea, that. Right, you can do something like this in order to summarize those 2 processes. Yeah, what happens in pollen formation, what happens in ovule formation, and what is common between the 2. You could also go further if you were a bit more confident and do a comparison between gamete formation in plants and gamete formation in animals as well. Okay. Similarities and differences. So one of the main thrusts of A-level questions is they like to go across units. Okay, so very rarely do you get a nice little neat question on plant reproduction. Okay, there'll be bits from elsewhere on other units. Okay. Sorry, excuse me. Right then. Okay, so pollination, which is the plant version of fertilization, which is the movement of the male gamete to fertilize the female gamete. Okay, so pollination, the transfer of pollen grains from the anther to the mature stigma of the same species. That's a slightly better definition than what I've just said there. All right, so self-pollination and cross-pollination. So self-pollination is where pollen will transfer from the anther of a flower to the mature stigma of the same flower or another flower on the same plant of the same species. Okay, so that's all within one plant. Okay, it can either be within the same flower or it can be on a different flower but on the same plant. Okay, cross-pollination is transfer of anther from a flower to another flower of the same species. Okay, so cross-pollination, different plants, self-pollination, same plant. Leads to self-fertilization, which leads to inbreeding and outbreeding. Okay, inbreeding leads to less genetic variation. Okay, outbreeding leads to more genetic variation. Okay. These are the cellular processes that result in that genetic variation. Okay, so combining of genotype from 2 different plants that obviously leads to genetic variation. These 3 things here, okay, are methods of of gaining variation at a cellular level, at a chromosome level. Okay, so again, here there's a synoptic element because they can ask you questions about 1.6 from last year. Yeah, not specifically all about like cell division, but these things here you need to be aware of and remember from last year. Okay. Greater chance of two potentially harmful recessive alleles combining, which is basically what inbreeding is. Okay, and outbreeding, there's a lesser chance of that. Okay. The advantage, okay, for self-pollination is successful plants can go crazy. They can just breed and breed and breed, and that's fine. And in a stable environment, that's brilliant. Disadvantage of that is if the environment changes, they have a very narrow genetic pool, yeah, a very shallow genetic pool, so they haven't got the genetic resilience in order to survive. Okay, cross-pollination is kind of the other way, yeah, so advantageous in terms of evolution, because if there's environmental changes, the chances are they're more genetically diverse, more genetically resilient, they'll be able to survive. Okay, so but yet again, you know, you can draw comparisons here between pollination, between fertilization as well. Yeah, you can maybe do one of those Venn diagrams to summarize that. Okay, adaptations of flowers to promote cross-pollinations. Okay, so chemical self-incompatibility. So that means that the gamete, if it happens, if the pollen happens to fall on the stigma of the same plant, they are chemically incompatible. Okay, so that means that fertilization won't happen and the seed won't develop. Irregular flower structure, so this happens in, as it can say, primroses here. As you can see, this is the thrumide variety. So these are the anthers, and in this one, these are the anthers. And I'm sure you can tell me which one is more likely to be self-pollinated. Yeah, this one. Okay, there's a greater chance of this pollen falling onto this stigma, isn't there? Okay, and there's a much less chance of that pollen falling onto these stigmas. Okay, so this is more likely to be cross-pollinated. Dichogamy, where anthers and stigmas mature at different times. Yeah, so you have, so that will reduce the chance of self-pollination of pollen falling onto onto the stigma of the same plant. You also have monoecious plants that have separate male and female flowers. Okay, so that happens on hazel. So, you know, in spring, sometimes hazel trees have little lamb's tails. Okay, so they're male flowers. Okay, and they will mature at different times to the female flowers on the same plant. You also have dioecious plants that have separate male and female plants. Okay, so if you've ever seen holly with berries, that is a female holly. Okay, and holly that has little, like, they look like little trees on them. Okay, that's male holly. Okay, so they have different completely, so they can't self-pollinate. Okay, so promotion of cross-pollination, 5 different methods there. Okay. Fertilization in plants, slightly more complex than fertilization in humans because it is double pollination. Okay, so this involves 2 separate fertilization events. So the formation of a diploid zygote, okay, similar to humans, and the formation of a triploid endosperm nucleus. Okay, so this is basically forming like a store of food for the seed. Okay, but we'll come to it in a minute. Okay, so this pollen grain has landed on this stigma. Now, whether that's wind pollinated, insect pollinated, or whatever, okay, doesn't matter. But remember, in here we have 2 nuclei. Okay, so we've got the generative one and the tube nucleus one. Okay, so the pollen tube will grow down through the style under the control of the pollen tube nucleus. So this here, this nucleus, will code for the production of enzymes which digest a path down here. Okay, so this pollen grain will germinate and it will send a little tail down, that's what this white thing is, down here to this. Okay, So during this, okay, you've got the tube nucleus is merrily mining its way off towards the ovule. Okay, during this, the pollen tube will divide into two gametes. Okay, so as if it wasn't complicated enough now. Okay, so instead of there being two here, we've now got three. We've got the tube nucleus, and then the generative nucleus is split into two nuclei. Okay, so that's another mitotic division. Okay, right, so number 4 here, the pollen of the tube is positively chemotrophic. Okay, so the pollen tube grows towards chemicals secreted by the embryo sac. Okay, so the nucleus, the tube nucleus produces the enzymes. Okay, and it grows in the direction of the embryo sac there. Okay, right, the pollen tube grows through the gap in the integuments. Okay, so there's a little hole here called the micropyle, and that pollen tube nucleus grows, got that weird line again, grows up through into the embryo sac there. Okay, once the pollen tube does that, it's done its job, it degenerates. Okay, right, so we now have, okay, so now you're probably wondering, double fertilization. Well, we've got two nuclei here, haven't we? Yeah, so one of the nuclei, sorry, will fertilize the male gamete. Okay, so one of these will fertilize the female gamete. Okay, and that will become the embryo. Okay, that will become the diploid zygote. The other one, okay, let's do that in a little box to be different. That will fertilize the polar nuclei. Okay, and that will make a triploid endosperm nucleus. Okay, so after fertilization, after our double fertilization, okay, these antipodal cells and the synergids, no further part, end of. But what we've ended up inside our embryo with is a diploid zygote and a triploid endosperm nucleus. Okay, so that is a very complex process. Okay, and there's lots of steps there. Okay, so you need to learn the sequence and what happens at each step. Right, the diploid zygote divides by mitosis to form the embryo, which will then differentiate into the pre-shoot, which is called a plumule, the young root, the pre-root, which is called the radicle, and one or two seed leaves, okay, that are called cotyledons. Okay, excuse me. So in this seed here, okay, this is the micropyle. Okay, so if this is the seed that's grown from the fertilization we've seen before, okay, this is the micropyle. So that female gamete was here, that's been fertilized, and that has grown through mitosis to produce the plumule, okay, and the radicle, okay, and there's going to be two cotyledons here. Okay, so when that seed germinates, it's going to have two little leaves of its own. Right, the endosperm that I have promised to tell you about. So the endosperm tissue divides by mitosis to form basically a food source. Okay, so that triploid cell in the middle is going to develop into this brown thing here. Okay, right, so these type of seeds, okay, so these are endosperm seeds, they have that store of energy there. Okay, now in these seeds, in dicotyledons, what happens is that endosperm is absorbed into these cotyledons. Okay, so what that means is that when this seed germinates, it's going to use all this store of energy here, this store of starch, to grow its plumule and to grow then a leaf when it's out. Okay, this here is going to grow, use the endosperm that's in the cotyledons, and two of those leaves will two of those leaves will be present when the seed is germinated. Okay. So the seed here, that has developed from the ovule rather, sorry. Okay, so the ovule develops into the seed. The integuments, you remember the two protective membranes, or the two protective bits of tissue around the ovule, produce the tester, okay, which is the seed coat. So that's the hard protective seed on the outside. The micropyle develops into that tiny little hole there. Okay, so that's how water gets in. The ovary wall, okay, so you've got lots of these inside the ovary. That ovary wall develops into the fruit. Okay, so you know, if it's an apple, the pericarp, the fruit itself, will develop from the ovary wall. Contents of the ovary is the fruit, and the attachment point is the hilum. Okay, so where the funicle, that will develop into something called the hilum. Okay, so not only do we have complex double fertilization, we also have afterwards different mechanisms for survival and growing of seeds depending on whether we're a dicotyledon or whether we are a monocotyledon. Okay, so different species have different mechanisms. There are also different mechanisms of spreading seeds as well. Okay, so some seeds have some seeds are wind spread, some seeds are spread by animals, some seeds are spread by the pods exploding, different things like that. Okay, so seeds spread by animals tend to be inside tasty things. Okay, so fruit seeds, for example, will be inside a fruit. Animal will eat the fruit, it will fly or walk far away, and then it will deposit the fruit with its own little dollop of fertilizer. Some seeds that are wind dispersed, okay, so things like dandelions, yeah, they've got the little like the little cotton woolly bit on top, and that's blown on the wind. Okay, so different seeds have different mechanisms of dispersal. Okay, so seeds can go through a period of dormancy, okay, which means that basically, don't use the word asleep, okay, but they're in a dormant state, they're not ready for growing yet. All right, that enables them to wait until conditions are right to grow. Okay, so seeds will require water, oxygen, and a suitable temperature in order to germinate, and unless they have those three things, germination won't happen. Okay, so they require water in order to mobilize enzymes inside the seed so they can travel, so those enzymes can travel into the endosperm, start breaking it down, start releasing the glucose from the starch, so the seed can then carry out respiration and grow. All right, oxygen for aerobic respiration, as I've just mentioned, and then suitable temperature again for the enzymes to operate. All right, so germination involves a rapid onset of biochemical activity. Okay, so that water goes in, it activates those enzymes, and those enzymes move around the seed, and then there's a big biochemical party happens. Okay, right. They do quite enjoy this graph, the question writers at WJC. So this is mass of the seed, okay, or mass generally, and this is time, okay, and these are three different things. Okay, so this is the mass of the endosperm that we just talked about. This is the mass of the embryo turning into the seedling, and this is the total mass. Okay, so hydrolysis of food reserves, okay, so that converts the starches into sugars. There's a reduction in mass there. Okay, so the endosperm is getting lighter, and therefore the seed in its entirety is getting lighter. Okay, dry mass of the endosperm cotyledon decreased because carbon dioxide lost when the sugars are used in aerobic respiration, so CO2, and the sugars are sent into the embryo. Okay, so the weight of the embryo is increasing because sugars are sent there and mitosis is occurring. Mass is increasing. All right, the total mass, okay, will decrease because of what I've just mentioned there, but the mass of the embryo is increasing because the sugar's moved into it. But at a certain point along here, it will start carrying out photosynthesis. Okay, the seeds germinated, particularly with a dicotyledon, it will have those seed leaves, these cotyledons, and they will begin to photosynthesize, and the mass will carry on going up and up and up. Okay, so this is quite a neat way of explaining lots of things, really. Okay, so an annotated graph is quite a neat way of explaining what's happening there. Okay, so what's happening, why is it happening? Okay, so again, kind of like a structure and function deal. Yeah, what's happening where? Excuse me. There is also another specified prac that they like to use, and it's investigating the digestion of starch using the germination of seeds. So what you have is starch iodine stained, starch agar stained with iodine. Okay, so all of this bit here is blue-black, okay, because we've got starch plus iodine all over here. All right. An unboiled seed is put there, and if that seed's germinating, it's producing enzymes, so it will produce some amylases. And those amylases will break down the starch, which means my iodine and starch agar is no longer blue-black, but it's then clear. Okay, so there's no starch around here because the enzymes produced in this unboiled seed have diffused, is the word I'm looking for, diffused out into the agar. Okay, this one, boiled seed. Okay, boiled seed, enzymes denatured, so no enzymes diffusing out into agar, so no breakdown of starch, so it remains blue-black. Okay, and again, yeah, there's another synoptic element here, isn't there? There's a little bit about enzymes that we've talked about, so the hydrolysis of enzymes in the seeds, and then the enzyme responsible for the digestion of this starch as well. Okay, they do like asking this as a scenario, as a kind of what happens here, what happens there sort of deal. Okay. Excuse me again. So germination of our non-endospermic seed. Okay, so these are dicotyledonous seeds. Okay, so water is imbibed, which is a fancy word for saying absorbed. Okay, so water goes into the micropyle. The cotyledons will swell. The testis splits open, allows more water and oxygen in. Starch and protein reserves in the cotyledons are hydrolyzed, used as a source of energy then for this to grow. Okay, so these dicotyledonous plants, it's very important that they aren't planted too deeply. If they're planted too deeply, their reserves of starch and protein are going to be used before the leaves reach the surface and can therefore carry out photosynthesis themselves. Okay, so that is the difference there. Okay, that is basically what I've said before about the germination of an endospermic seed. Okay, so that's a monocotyledon, what's happening there. So again, more imbibing or absorbing of water. Gibberellin, so that's a plant hormone to do with growth, released into the embryo. Gibberellin diffuses into the alurone layer. So the alurone layer is the layer on the outside here. Okay, so what happens on the alurone layer is the production, then proteins broken down to amino acids into amylase. Okay, amylase breaks down starch into sugars, and then those sugars will then allow that monocot to grow. Okay, so this will have one seed leaf, monocotyledon. Okay. Excuse me, there's a frog in my throat. So again, here there are two different sequences of events that are occurring for those different types of seeds. Okay, so it would be good if you would be able to draw a comparison, either a Venn diagram or a table of what's happening in both different types. Okay. Right, okay, so that is basically the content for the unit. Okay, what we've revised up to now has been recall, okay, and consolidation of subject knowledge. So making sure that you are make sure all the information's in your head, basically. Okay, you probably know application type questions can be more difficult. Okay, so you need to have the subject knowledge at your fingertips, so you are then able to apply it to different situations. So what we're going to do now is a little bit of practice. Okay, so the following slides are a mixture of past paper questions for you to try yourselves and some peer marking exercises. Okay, what I like to say usually to my students is 50% of the time should be recall, consolidation, making notes, and the other 50% should be answering past paper exam questions. Okay, so first question here, what I would like you to do is stop the video, have a go at answering those questions, and then in a minute I will show you the mark scheme. Okay. There's another part to this question just there. Okay, so you can stop the video again and write the answers down on a piece of paper, and in a minute I will show you the mark scheme. Okay, so the mark scheme's coming up just now. Remember, when you're using mark schemes, okay, anything that has wavy brackets, such as variation here or diversity, that is a requirement, okay? ORA means or reverse argument. So you can say the stigma above the answers, or you can say the answer, yeah, you can use the other argument the other way around. Okay, so mark yourselves there out of 8, I think, or 7, I think. 7. Good job. Okay, next question. 4 marker now. Okay, so again, lots of the stem of the question is quite large here. So stop the video. I would like you to read through it. I would like you to highlight the command words. What is it asking you to do? Okay, and then what do you think are the key words? What information do you need? All right, this is quite a nice question because it has a describe. Okay, describe is literally say what you see. Okay, how would you do it? Okay, so how would you do that practical? Good job. So the answer is coming up now. What students sometimes don't like to do is do independent thinking. So, for example, that experiment there asked you to basically think about and design an experiment. Okay, you know, any method is okay as long as it's answering the question. Okay, so you need to be confident in about you know, what am I looking for? What else do I need to keep the same? You know, what's the independent variable? What's the dependent variable? Yeah, what are the control variables? Okay, right, next question. State and explain the conditions required for germination. Describe the germination of the peanut and barley seeds shown above. There is a lot to unpack here. There's loads of marks available here. State and explain. So say what and then say why. Okay, what and why. And you've got barley, okay, and you've got a peanut. So one is an endosperm and one is a dicot. Yeah, so this is going to be the leafy one, that's going to be the stuff drained, and this is basically just a learny one. Okay, right, I'm going to shut up now and let you have a go at that. So stop the video, and in a minute I will produce the mark scheme for you. Okay, so loads of marking points there that you could remember. Okay, right, next question coming up. Okay, so again here, okay, we've got a state. Okay, so they're just recall questions. We've got 2 explains here, so we've got 2 say whys, and we've got a describe. Describe and explain. Okay, so one say what you see and one say why you'd see it. Okay, so in a minute So stop the video, write your answers down, and then in a minute I will produce the mark scheme for you. Okay, so here's the mark scheme for that question there. So stop the video and you can go through those questions. Okay. Right, one last one for you to do independently. Okay, so there is a photomicrograph, so that's your tapetum. Okay, so state the function. What is the job of? Explain the appearance. That line again. Explain the appearance of the tetrad. Okay, and then describe the function. Okay, so describe ones are nice. It's just say what you see. Yeah, describe the function of the tube nucleus. Okay. Right, so you can stop the video and write those down on a piece of paper, and in a moment I will produce the mark scheme for you. Okay. So mark scheme being produced now. No, it isn't. Afraid. Sorry, there's another part to that question. Okay, so you can stop the video on this bit and answer this bit, and then in a second I will produce the mark scheme for you. Okay. So mark scheme being produced about now. There we go. Big question, that one. Okay, lots to get your teeth into. All right, and a mathsy one in the middle. Very good. Okay, so again, synoptic element microscopy from year 12. Okay, good job. So stop the video there and do your corrections. Right, next one is a peer marking exercise. Okay, so I'd like you to, this is the question that you've just answered. Okay, so you can go back to the mark scheme for this. Okay, you can scroll back through the video and find it, and I would like you to think, I would like you to give Johnny how many marks you think he deserves for those answers. Okay, how many marks Johnny got for those answers. So stop the video, or you can scroll backwards and forwards and mark his answer. Okay. So the next part of the question coming up here. So this is the other bit, the other third of Johnny's answer. So stop the video here and tell me how many marks you think Johnny got for that. Okay, so here are the answers. So anywhere on a mark scheme or on marking where there is one of these, okay, that means there's a word missing. Okay, so what word do you imagine that is from the mark scheme? Okay, food, that is far too generic a term to be used in A-level biology, I'm afraid. Okay, and then here, Johnny again has missed out a key term just here. Okay, oops, pardon me, missed out a key term just here. Okay, what's he missed out? And our Johnny obviously is a very good mathematician there. Okay. Next one, labeling. Yeah, got his labels right. Okay, so knows what he's doing there. All right. Meiosis happens in the megaspore. Meiosis 3 times develop gametes. So again, there's a lack of using correct terms here, isn't there? Okay, so Johnny needs to, and again, on this bit here, he's missing out keywords. All right, so again, you need to drill, make sure that those keywords are straight and square in your head. Okay, good job. Right then, another question. Okay, so this is for you to have a go at, and then we'll do a peer marking exercise on that. Okay, so you can stop the video just here, have a go at that. Other half of the question coming up now. Okay, so it's that question that they like asking about the embryos. Okay, independent dependent variable. Okay, so stop the video now and have a bash at answering that. Okay, so the mark scheme for that question being coming up now. Okay, so not a terrible question, that one. Okay, so there's some good questions there for you to get your teeth into. All right. So now we're going to do the same thing again. If you have marked your first thing there. Okay, so here's Johnny again, and he's answered this question. So I'd like you to scroll back into the back in the video to the mark scheme, and you can see how many marks you think Johnny deserves for his answer. Okay, and second half of the question coming up now. Okay, so those are Johnny's answers. So if you stop the video here, scroll back to the mark scheme, you can see how many marks Johnny got. Okay. Good job. Okay, Johnny's actual marks coming up now. Okay, so 2 marks that first one. Okay, got his marks for the description of the wind and animal dispersed seeds there. Okay, nothing going on here. Okay, there you go. Sometimes you just get wrong answers. Okay, but I'd like you to think, how could he change that answer in order to squeeze a mark out of it? Okay. Second bit now. Okay, what marks did Johnny get here? Okay, so you got 1 mark there for that. Size of the embryo, again, that's not quite the level of detail we're after really for A level. Yeah, size of embryo. You have a look at the mark scheme and see what they're offering there. Okay, and then only 1 of the 3 here. Okay, so some of the common mistakes that students make are yeah, lack of detail. Okay, it's either lack of detail because you haven't revised enough, you don't know the answer. Okay, it's lack of practice, so you're not understanding what the question's asking, or it's how you word your answer. Okay, so those are the 3 things that you need to make inroads into. Okay. Right, so over to you. We've just done a little bit of practice and revision here, and there are many different ways of revising. Remember, you need to recall the information and answer old paper questions. You need to make sure that you're doing that application work in order to get the grades. Okay, so the important thing is that you need to work hard. Remember, there are lots of resources available on Cadlam Cymru, and all the very best for your exams this summer. Okay, grand. Thank you very much. Hwyl fawr. Ta-ra.